Good morning, everyone. I hope you can hear me. So my name is Arun Mistry. I'm one of the physicians at Minoryx Therapeutics. And thank you very much, Elisa, Miranda, ALD Alliance, for inviting us to provide an update as to where we are with our program. Very exciting to see everybody dressed in green as well. So I think that's fabulous to see as well. So congratulations to everyone. So, a few updates in here, which I think will be new for many of you for the first time. And some of this is old news as well, but just to refresh. So Minoryx, we're a small, late-stage orphan drug company. We have one product, leriglitazone, which we see potential for many different CNS disorders. Really, rare diseases with high unmet medical needs is where we see the value of this product. The product is from a drug class called PPAR gamma agonists, and really comes from a family of type 2 diabetes drugs, pioglitazone, rosiglitazone from the past, but it's from that family. So it's quite a well-characterized molecule in that sense. But one of the differences that we see with this product is that it's brain penetrating. So it actually crosses the blood-brain barrier much more than older products, which are of the same class. As an update as to where we are with the product across the multiple areas that we're looking, first of all, for XALD, we actually have the product in review for a marketing authorization with the EMA. So that's in progress at the moment. And we will hear sometime over the summer if the EMA grant an approval for the product. In the US and also globally, there's a pathway for XALD with the CALIX study, which I will cover. I think Dr. Fatemi mentioned the CALIX study, so I'll take you through that as well. But again, that's the pathway for the US. And then also we're looking at the product performing a phase 2A study in Rett syndrome, another genetic disorder typically seen in girls with developmental delay that starts from the age of 2 years onwards. As I've said, the product is brain penetrating. The other important point for this product is it's just an oral suspension. So it's not an injection, it's not anything too difficult for the patients to take, especially pediatric patients and adults. It's this oral suspension for all patients. We see it as a disease-modifying agent, and I'll take you through the different modes of action in the next slide. But with this different characterization for this product compared to the other PPAR gamma agonists, we've been able to take this to the regulatory bodies, to the EMA, to the FDA. We have orphan designation with the EMA and FDA, which gives us a more rapid pathway to be able to access for advice and help. We have fast track and rare pediatric disease designation from the FDA, again, giving us more access to help and advice. And this is how we got the advice on designing the CALIX study as well. In Europe, we have the product out-licensed to a company called Neurax Pharm for commercialization. So Minoryx, as a company, we are 30 people globally. So we're very small. We're not going to be able to launch the product and commercialize, so we look for partnerships. And then also in China, we have the product out-licensed for future once it's approved through a company called Spirogenics. So just a little bit more detail on the mode of action and how we see, of course, specifically in ALD, how the product can help, how we see that working through the preclinical and laboratory type of data, but then also we're getting signals from the clinical trials as well. So one of the key aspects, and this is where the rest of the talk will really sort of focus, is an action on neuroinflammation. So cerebral ALD is really an inflammatory process. Once the blood-brain barrier is broken, there's a cycle of inflammation that goes on inside the brain, which increases the size of the lesions and continues the breakdown of the blood-brain barrier. What we see with the product from lab data is that there's a reduction in inflammation, particularly acting on the white cells in the brain, so the macrophages, the microglia, which is the inflammatory cells in the brain itself, and also on the endothelial cells, which are the cells that make up the blood-brain barrier, and really sort of reducing the inflammation there. Also, we see from our experiments pro-inflammatory mediators being reduced as well. And with all of that action, we actually see levels of improvement of that blood-brain barrier integrity. So we see preservation of that integrity when it's not broken. Another effect of the product is on myelination. And this is where, as you've seen, there's demyelination that happens in the brain, but also with ALD, of course, we have AMN, adrenomyeloneuropathy. And this is an effect probably more so for the spinal cord, where we really see a reduction in demyelination with the protection of the product with the PPAR gamma agonist effects on the oligodendrocytes and improving the way that myelination is maintained, and also removing any myelin degradation products from the CNS, which can again cause further inflammation. And then the final mode of action is really improving what we would call the mitochondrial system, and that's the energy supply in all cells. But what we see with the product is really improving that energy balance, reducing what is called oxidative stress, and this can be happening again, particularly in the adult AMN patients, and reducing the axonal damage. So again, working on the myelin, but also improving the energy balance in systems. And elements of this all contribute to sort of neuroprotection of the brain and the spinal cord. And that's how we see it in ALD, but also all of these different modes of action can help in other diseases, and that's why we're looking at the Rett syndrome study as well. So, leraglutazone in ALD. Very briefly, I think you've seen all of this before. ALD is an X-linked genetic defect in ABCD1. The focus of the rest of this talk is really on CALD, on the cerebral form of the disease, which affects boys and men, the demyelinating lesions in the brain, and then, of course, the progressive nature that can happen, which has been explained again by Dr. Fatemi and Dr. Raymond just now as well. But there is the AMN element of the disease, which causes spinal cord disease in adulthood, affects men from the age of 18, 20 onwards, and then also affects women who were called carriers, but they're affected, and affects them later in life, but they do have problems with their mobility. And I think, again, Dr. Fatemi explained other elements of ALD that are seen now within the data sets for natural history in women. Importantly, cerebral ALD and AMN can coexist and develop independently. So it's not sequential. It can happen at any time in terms of CALD. So I'd like to introduce you to a sort of different graphic, again, just the evolution of how the thinking is going for how lesions evolve in the brain. So first of all, you've seen the MRI scans with the typical less score that Dr. Raymond described. But there are even more, I suppose, interesting sort of experimental ways of looking at MRIs with what can be called diffusion tensor imaging, perfusion imaging with the MRIs, and they can pick up non-lesional effects, which can be seen, but the less score can be normal. So no lesions, but the MRI can still look abnormal. And, again, it will be something for Dr. Fatemi and Dr. Raymond to comment on in terms of what these other differences could mean, but the focus still these days for routine care is lesions and the less score. So if there's no visible lesions, then continued screening, as Dr. Fatemi said. But as soon as there is a lesion, then it's monitoring. And the first lesions, then there's increased monitoring. The lesions often start gadolinium negative, so non-enhancing. They turn gadolinium positive. When they do that, they start to grow much more rapidly. But, as was being discussed before, gadolinium negative lesions can grow rapidly as well. So, again, acute monitoring is very important. These lesions progress and can, over the course of 2 to 3 years, progress very rapidly. They're monitored with the less score, with lesion volume, as has been explained again, the neurofilament light chain plasma results as well can be used to monitor. So all of these things, some of them are still under research, but the less score is the key tool for monitoring the progression of lesions. These biomarkers often precede any clinical symptoms. So clinical symptoms only occur when there's damage ongoing in the brain. So clinical symptoms like the neurological functional score or major functional disabilities, which are the way of monitoring, especially in pediatrics, they happen after lesions are already identified typically. And then just sort of looking at the numbers of patients, and this is fabulous to hear that there's this fantastic epidemiological work ongoing from the newborn screening. And this will really help us as well, helping understand where patients are and how they can be helped in the future. So we think from estimates from historical publications and epidemiology that there are probably about 7,500 patients in Europe and the USA with brain lesions, gadolinium negative or gadolinium positive. From these 7,500 patients, probably 2/3 of them will develop progressive CALD, 1/3 of them during pediatric age time, and then 1/3 during adulthood, which, again, as Dr. Fatemi explained, could happen at any point from the age of 18 to 65, 70. Less so in the older age groups, but can happen any time. And the standard of care is transplant for progressive disease. This is invasive, risky. The data from publications is that it does lead to a high mortality and morbidity for the patient, so it is a risky procedure. Not always widely available, and finding a donor can take time as well. In the adult patient population, it is happening more frequently for progressive disease, but again, because of the comorbidities of AMN in the adults, there's a poorer survival and a poorer outcome in the adult population. In the US, of course, you've got gene therapy, but it's only available here in the US. In Europe, it's not available. So standard of care globally is transplant. If a donor is not available, then gene therapy in the US. But there are also no treatments to avoid progressive disease. And this is where we sort of start seeing potential for some of where our data is starting to take us from our clinical trials. So I'm going to take you quickly through 3 data sets. And thus far, we've got data on more than 150 patients with cerebral ALD who've received liraglutide. So the first data set and new information for for you today will be the confirmatory analysis for the Nexus study, which is a pediatric study in CALD. Then a very brief review of the advanced data, which is a study in AMN, where we were looking at the 6-minute walk test, which didn't succeed as a primary outcome, but we saw some very important signals for how disease progression for CALD occurs as well. And then finally, some data on compassionate use. In Europe, we have a compassionate use program for liraglutide in CALD. Be able to take you through some of that information. We have more than 100 patients treated in Europe thus far for compassionate use, where lesion growth and disease arrest is being monitored. We have a study that will present from France. But in addition, there are 2 cohorts in France and Germany where we've been able to collect data for the authorities for safety and efficacy, just to show them that the product is providing some level of benefit and value for the patients prior to any approval. So firstly, the Nexus study. This was a phase 2/3 study in boys with cerebral ALD. It was open label in design, and really using natural history from publications to state what the natural history of a self-arrest rate would be in terms of a disease being identified, lesions being identified, but then not progressing. The study enrolled boys aged 2 to 12 years old, both with gadolinium negative and gadolinium positive lesions. The LES score had to be less than 10, but they had to have lesions. And neurological functional score less than or equal to 1. We enrolled 23 patients in total, and we had 20 evaluable patients in terms of patients where we could check on how they were doing compared to baseline with the appropriate assessments on the MRI scan. The study was organized from being 96 weeks long, and we now have data longer than 96 weeks in some patients now, but the primary analysis was at 96 weeks. We had a continuation analysis at week 24, just to make sure that we were seeing some level of effect to continue doing the study. The primary endpoint was the rate of disease arrest, and again, looking at the historical publications. But then we also had some secondary endpoints looking at the LES score, the clinical elements of the neurological functional score, the MFDs, the gadolinium intensity score, survival, and also patients who met transplant criteria. Some exploratory endpoints were looking at the lesion volume, and this is work specifically done together with Dr. Malak when he was in New York, but now also in Baltimore. And then we have some plasma biomarkers as well. Important to state that this study was designed as using liraglutide prior to transplant. So if patients needed a transplant, they could go to transplant at any time point according to the physician's decision. But the study had some criteria, really just for an analysis basis as to patients needing to go to transplant. And this is the study that's being used for the EMA filing. So back to that diagram again, and then really just sort of explaining the natural history studies. So these are publications, several publications, which look, and it's difficult to do this now, look at how patients would behave if there was no treatment available. So no treatments available for gadolinium negative disease or gadolinium positive disease. And most of the papers really sort of lead to a self-arrest rate whenever lesions are found, gadolinium negative or gadolinium positive, of about 10%. So that's one thing that we used as let's say the control for the analysis. What we saw was, first of all, looking at the populations of gadolinium negative and gadolinium positive. We had 9 patients enrolled into the study who were gadolinium negative, so before enhancement was seen, and 14 patients who were gadolinium positive. What we saw for the whole group together was a disease arrest rate of 35% compared to the 10%. So this gave us a primary endpoint outcome which was significant for statistical basis, but also which really led to the study being reviewed by the EMA as well. This disease arrest was composed of both clinical and radiological elements. So the NFS had to remain stable, no major functional disabilities, and also the level of lesion growth had to be reduced compared to what natural history would be as well. So to break some of that down, first of all, looking at the neurological functional score. And this is the paper that's been referred to previously again by Dr. Fatemi, Dr. Raymond's paper on the natural history. So neurological functional score is the typical assessment in pediatrics of understanding how clinically there is worsening without treatment. And here you can see the neurological functional score worsening in terms of having a higher score with time. And this is both for gadolinium negative and also gadolinium positive patients. You can see the 0 down at the bottom, and that's the time point of gadolinium positive disease being identified. So patients seem to progress quite rapidly. Again, time is a context that we need to sort of take into account, but this is quite rapid neurological progression that would be seen. So that's natural history. With transplant, and this is in patients who are gadolinium positive. So again, making sure that we place this in context. These are patients who are gadolinium positive at the time of transplant. And you see a stabilization, just very little worsening or increase in the neurological functional score. And then when we look at all 23 patients from Nexus, and all 23 patients are on this graphic, there was little or no increase in the neurological functional score. Either stayed at 1 or stayed at 0. There was 1 or 2 patients that went from 0 to 1. Of course, this is data that also includes patients going to transplant. So 14 patients did go to transplant out of here. But we do have 8 patients who remained on study past week 96. So this is all of the data. And then, of course, we've got to look at the LESS score. And again, when we look at other therapies, gene therapy and transplant, what happens to the LESS score once you start having the intervention and treatment? As Dr. Raymond said, most of these LESS scores do remain fairly flat. There's some worsening in transplant and also in gene therapy, but typically these are maintained low. And again, we see something similar here with the Nexus data. And again, you'll see the patients being presented here is split between the GAD positive and GAD negative. So the GAD positive patients are in red and GAD negative patients are in blue. And you see there are some patients with a worsening of the LESS score, and typically these patients went to transplant quite quickly, which is part of the treatment paradigm that was set up within the design of the study. But many of these patients, up to the point of transplant, were not having large increases in the LESS score. So we see some benefit there in terms of maintaining the lesion load to as small a level as possible prior to transplant. And then moving on to the advanced study, and this is data that I think you've probably mostly seen before. This was a study in adult men with AMN symptoms, but patients were enrolled with and without lesions at baseline in terms of their cerebral lesions. And 30% of the patients had lesions at baseline. The study was a double-blind placebo-controlled study with a 2-to-1 randomization to liraglutide. So we had 116 patients going into the study with 39 on placebo, 77 on liraglutide. And that double-blind period was up to 96 weeks, and then there was an open label phase. The primary endpoint was 6-minute walk test, which unfortunately we failed on. Probably not the endpoint or the variable to use in this very slowly progressing part of the disease. So some more work needed there. We did see some good signals for body sway, and I know there's a lot of work ongoing globally for body sway and how that can be used as an endpoint. But we did have some predefined variables for CALD. So we had the incidence of cerebral lesion progression, we had the MRI less score as well, and then we also had the neurofilament light chain plasma levels being assessed. In addition, the clinicians at the centres were also diagnosing progressive disease, progressive CALD, because all of these men coming into the study did not have progressive disease. They just had, in a sense, AMN, maybe with lesions that were not considered to be active. So again, going back to that figure, so we had patients just in that second column of, well, first and second, no lesions or some GAD negative lesions. And what we saw from the safety data was only patients in the placebo arm, 6 out of 39 patients developing progressive disease. No patients in the liraglutide arm. So this was safety data. It wasn't an endpoint, but gave us a clear clinical signal that something was beneficial when we look at this data in this way. And when we looked at the CALD parameters specifically, which were part of the study design, we saw in terms of cerebral lesion progression, again, only 3 out of 77 patients on liraglutide progressed or had some level of lesion progression, compared to 8 out of 39 on placebo. So again, statistically significant, and we feel clinically of relevance to think about as well. And this was reflected again in the less score, where the less score increase on liraglutide was statistically lower than those on placebo. And then finally, for neurofilament light chain, there was an increasing in the neurofilament light chain plasma levels at week 96 on the placebo patients compared to the liraglutide patients. So again, something that's very important for the adult population that we will be sort of taking forwards. And then finally, in terms of data, as I was saying, we have a compassionate use program in Europe. In France, one of the things that the French clinicians did was collect the data more formally, and they published this data in 2024. And this was after a 2-year follow-up of these patients. They had 13 patients that they were following up, aged 19 to 67. 10 out of these 13 patients who were diagnosed with progressive CALD, some GAD negative, but mostly GAD positive. 10 out of the 13 remained clinically and radiologically stable at that time point of 2 years. 4 out of the 10 were stable clinically, and also some of them actually had some level of improvement in their clinical status or neurocognitive status. There were 5 patients who were listed at that time point of 2 years, well, at baseline for transplant. All 5 came off that transplant list at 2 years. Of course, further data will need to be assessed as to what happened to them afterwards, but in terms of that 2-year effect, we see something as a signal there. And then we also saw gadolinium enhancement resolution in a certain number of patients as well. So we were starting to see some other signals of how the product could be affecting adult CALD. Of course, this was not a clinical study, and that's why we're doing the Calyx study, which I will cover very briefly as well. But importantly, just to think about the safety aspect as well. And this is something that we are, collecting very logically, but also with a lot of diligence as well. But we have, in total, about 450 plus years of exposure in adult patients, 116 plus years of exposure in pediatric patients. In adults, there's a lot of expected edema, peripheral edema, so edema of the lower limbs, and weight gain. And these are known effects of PPAR gamma agonists. So pioglitazone, rosiglitazone, these older products led to these sorts of things. We see it more frequently, the reason being we have more affinity for the receptor than the other products. So it is seen in adults, probably about 80% of patients will get some level of edema, but we don't see that as causing any cardiac dysfunction. So again, we don't see that fluid retention causing any issues. Pediatrics, there's less edema because the boys are much more mobile, running around. But we do see weight gain, not reported as adverse events, but when we look at the weight gain charts, they do, after 6 months, have an ongoing increase. The height stays at their expected levels, but their weight does go up. But again, no cardiovascular issues seen there. So we have a lot of safety data, which the EMA has really, in a previous discussion, said it was proportionate in a CALD population. So just finishing with the Calyx study, and this is the key pathway for the product being reviewed by the FDA. So this is a phase 3 study that's ongoing. We're looking here for men with gadolinium positive lesions who are not recommended transplant or not wishing to undergo transplant. We're looking for approximately 40 patients, and patient number 36 is randomized, so we're looking for just a final few more patients. We've got sites here in the US, Argentina, Brazil, Europe, and also India. Unfortunately, because, as Dr. Fatemi said, the natural history of adult CALD is not very well understood, the FDA really pushed us towards advised and then said, this is advice you have to take, to a placebo-controlled study, which is, in a rare disease, is something we didn't want to do, but it's something that we have had to implement. The primary endpoint is time to death, or the patients being bedridden with a need for permanent ventilatory support, and the key secondary is the LESS score. But this is a study that will take us, hopefully, towards the FDA reviewing for an approval in the future. So the study design looks a little bit complicated. We have a 1-to-1 randomization, liraglutide to placebo, so approximately 20 and 20. When the last patient, so hopefully in the next month or two, when the last patient number 40 or so is randomized, we're able to ask our data monitoring committee to look at the primary endpoint of survival at 18 months. If we have success at that point, the study stops, all patients transition to active drug, and we can then submit the package to the FDA. If that is not positive at that point, we can continue for another 9 months and repeat that again. If that's successful, again, everybody moves to active, and the study stops. Again, if not successful, we go all the way out to 3 years, 36 months, and conclude the study and analyze at that point. At each of these time points, typically the FDA will give us a statistical penalty for looking at the data. Even if the company doesn't look, if the data monitoring committee look, they normally give a penalty. But this time round, there's no penalty. We don't have to add extra patients or have a more stringent level of success. So this is the good news. It's much more like an oncology study. I'd like to thank ALD Alliance and the wider ALD community globally in helping recruit to this study. This is a study that started October 2023, and we're nearly at patient number 40. And it's with the help of everybody here, even with the newborn screening and genetic counselors. We've been trying to find patients, adult patients, through the cascade testing, through helping really energize the MRI screening across the globe. It's been fantastic as a big team effort. So really like to thank Elisa and Miranda for helping get us to this point, and the wider community. But it's something that we see as being valuable, will give us a definitive answer in some way, we hope, once we look at the analysis. And if we have the 18-month timeline being met, we will have an answer by the end of 2027. So 18 months from now, we may have an answer, which isn't too far away. So thank you all again, and I'll take any questions.
Been on the compassionate use study from the original advanced study, so it's going almost 8 years now. I mean, I've had no side effects for the most part, other than at the beginning, a little weight gain and edema. But since then, everything, I call it my strawberry drink because it tastes like strawberries every day. And if it's still going to be provided to me, I'm going to take it. But do we see it kind of as to be used as a preventative kind of treatment after obviously much more data, as if for children and adults, to take it with the hopes that it prevents cerebral ALD? Because I feel like right now when I'm on it, I was like, assuming I'm still going to continue to take it, and just to monitor is to see if I get cerebral ALD, which I have not yet. But I feel like that's the overall. Obviously, we want to help people now, but with newborn screening, we're eventually going to get to a point where the population is being monitored. They don't necessarily have cerebral ALD.
Sure, no, it's a very, very important question. We see signals from, let's start with the pediatric study. The majority of patients who remained, in a sense, in a disease arrest state were the gadolinium negative patients. Identifying early and treating early is where we see the signal for those patients. So 6 out of the 7 patients who met the primary endpoint were gadolinium negative and have not progressed to gadolinium positive disease. And that's 2/3 of the gadolinium negative population. 3 patients did have enhancement and had to go to transplant. But there's a potential for the product to be used as soon as a lesion is identified, gadolinium negative or gadolinium positive. If it's gadolinium negative, the signals are that the majority of patients could remain gadolinium negative longer term as a preventative. For adults, with the advanced data, again, it is that prevention of progressive disease is one side of things, but also prevention of new lesions growing. Again, they weren't the primary endpoint, so the regulatory bodies won't look at that, but they're giving us signals, and of course the whole data package will be submitted with the Calyx study in the future. So again, we see different avenues that we could approach with this product.
All right, thanks.
Thank you, Arun. It's nice to see you. So when you say that you're seeing it more in the GAD negative, how do we really know how many of those would progress? Because without newborn screening and monitoring, I mean, we see lots of people come in as teenagers or adults that have an old lesion that never progressed. But we don't know that. So we think that's naturally the group that probably is the least likely to progress. So I think it's hard to know what the rate really is for that group.
You're right, there's not as much natural history as to what happens to lesions once they're first identified, and we're actually setting up a study to look at that, to make sure that when we give that analysis to the regulators, they can look at it, comparing it to natural history. So that's number one. Number two, there is a signal, and this was presented, I think, by Dr. Fatemi again, from work that Dr. Malak has done in terms of when gadolinium negative lesions are found, typically within 6 months, 50% of those patients have gadolinium positive lesions. So we see that data, but we know we need more natural history.
Hi, Arun. Great talk. I'm Vinod Prasad from Genetics Biotherapeutics and Duke University. I want to congratulate you on this promising data and really able to recruit these many patients so quickly. So that's fantastic. I want to provide a little bit of clarification on the nexus comparison with the gene therapy. Every patient who was treated with gene therapy was gadolinium positive.
Yeah.
And that's what it is indicated for. It's indicated for early active cerebral adrenoleukodystrophy, and active means that it has to be gadolinium positive. So I just wanted to clarify that comparison between Nexus, which had a number of gadolinium negative patients, some had gadolinium positive, and then they probably also, some of them went to transplant. That the gene therapy data is all, and I think most of the transplant data is also
Yes.
all in gadolinium positive patients.
No, that's important, and I think I did mention that as I was presenting, that when we look at the LESS score and those data sets, they're not direct comparisons, we're not doing an analysis, but it's giving us a signal. And the key thing for the gadolinium positive patients is that we hopefully see, and we do see from our data, a reduction in the lesion load at the time of transplant, and that's something that we think is important.
It's a very nice talk. I wonder if a children has ALD, how long he need to take the pill to completely stop it? Did they need to take it lifelong, or just 2 years?
So the analyses that we've presented are for the week 96, 2 years, but the patients have, those who have, in a sense, not had a transplant donor available, they've continued on treatment, and those patients who are arrested have continued on treatment as well. So we see it as potentially lifelong to avoid the development of progressive disease, which could happen at any time in adulthood as well. So at the moment, the patients are still in those continuing are typically adolescents or in their teens. But we don't think that we see a time point in the study where we'll say, stop the treatment.
Thank you.
One more question at the back.
Was the drug specifically designed for ALD, or because I'm wondering about maybe applications to other inflammatory white matter leukodystrophies like Krabbe, or there's a couple others I know of.
So it's not specific for ALD. As you saw from the mode of action, we've got the anti-inflammatory, we've got the action on reducing demyelination, and then the mitochondrial activity as well. So if the disease has any of those elements, there's potential for using the product in those other leukodystrophies, or as we're doing in Rett syndrome as well.
I have my first boy, he was diagnosed at the age of 7. It was too late for transplant. But he's 19 and he's still with us. So will that be the last presentation you had, be something for him? We feel like he lost his abilities, but his mind, sometimes when my husband and I are talking, he goes, boo, like he's talking, he's reacting. And so we think that his mind is still there. And then we put movies that, he used to watch at 7 years old, and he remembers them, and he enjoys that. And so will that be something for him?
So we're not studying that type of patient population, but we have been, when patients who are not eligible for transplant because the less score is too high, those patients are being treated on a compassionate basis in Europe. If the disease is not active, then again, that's an area where we've not looked at. So it's only if the disease is active that we would, in a sense, see a potential. But again, that's something for the regulatory bodies to decide upon, and also the clinicians as well, as to how the product will be used. But we don't have data on long-term, let's say, arrested disease where there's already an impact. Okay, thank you very much.